So Delphine, that was a great sort of introduction to post-translational modifications, and we're specifically going to be talking about phosphorylation. And, not necessarily certain what people's kind of background knowledge was of post-translational modifications. We just wanted to take a second and give a little background on a few specific post-translational modifications that are seen with MDH. Sort of the neat thing with post-translational modifications is we covalently modify a functional group on the end of a protein or on the end of an amino acid. So you sort of have to have some kind of a nucleophile, generally nitrogen or oxygen, that you're going to be modifying in some way. And that allows you to change maybe the charge, maybe the size, the functional group, and so forth. And so we can see some common things that we see with malate dehydrogenase are methylation. And so here you can see sort of methylation of arginine. There are other amino acids that can be methylated. Here's acetylation, again, using acetyl-CoA and transferring that acetyl group to a nucleophilic nitrogen of lysine here to generate a post-translational modification. To sort of orient you as we go through this talk to some of the things that we're specifically doing is I've highlighted in red if it's something that we are specifically doing with our project. And there's not a lot of red that we'll see here, but there's a lot that we're going to talk about. And that also highlights this idea that there's lots of open space here for people to say, hey, I'm interested in studying this. And there's way more projects than we have time or people to sort of get through. So specifically, the post-translational modification that we're looking at is phosphorylation, right? And if you're not familiar with phosphorylation, you have serine, threonine, or tyrosine residues, less prevalently with tyrosine residues since they tend not to be as surface located. But we have a hydroxyl group that can be phosphorylated. We have a protein kinase for a terminal phosphate from ATP, that gamma phosphate from ATP, onto that hydroxyl group, generating a phosphorylated serine, threonine, or tyrosine residue. You can sort of turn off that modification by using a phosphatase enzyme. And so, as Celeste will talk about in a little bit, kind of making this modification is going to introduce charge, and that can have a lot of important implications. So as we think about here, how prevalent are things like PTMs that are phosphorylation in malate dehydrogenase, we can see that between both cytosolic and mitochondrial isoforms of MDH, that there's lots of phosphorylated serine residues that have been identified. There's lots of phosphorylated threonine residues. What's kind of interesting is there are some that are found between both cytosolic and mitochondrial isoforms for phosphorylated serine, but there's not overlap between the phosphorylated threonine residues. So some really interesting things to be able to study and sort of understand why we may have certain residues being phosphorylated and not. So that sort of leads us into thinking about, well, why do we have these post-translational modifications? What are some of these downstream effects? And ultimately, we're modifying that existing functional group on a protein, and that can have lots of downstream effects. It may affect protein-protein interactions. And again, this is something I am interested in, but this is not something that this project is specifically about. But you can basically look at for example, looking at the interaction of malate dehydrogenase with citrate synthase, right? So these are enzymes that are sequential in a metabolic pathway, so it sort of makes sense for them to interact with one another. And post-translational modification with phosphorylation is known to impact that. And I need to move my little, there we go. My little bar there was over my words. One of the things we are talking about is activity. So how can post-translational modifications sort of affect the activity of a protein? Whoops, there we go. And just as a simple plot here to show you, we're going to show a couple more plots that look like this. We basically can ask the question about how the relative activity of this enzyme is affected by the addition of some allosteric regulators. So in this case, we can see that compared to the control sort of situation with this enzyme where there's no inhibitor, adding citrate inhibits this isoform of this enzyme. And so these are some of the things that we are looking at. We're going to be looking at enzyme activity. And so I know this is a super busy slide, so don't take too much from this other than to highlight in these regions that we have circled here and highlighted sort of in yellow are 3 post-translational modifications that are of interest to us. Specifically, we're looking at serine 8, serine 45, and then a threonine 85. And here's why these are important, and again, how they can play a role in enzyme activity. So that serine 8 is important for binding NADH, which is the necessary cofactor that we need for malate dehydrogenase activity. And so we would expect that that residue is going to be important in that part of activity. S45 is important for dimerization of malate dehydrogenase. So again, another important residue that could play a role in regulating activity. And then that threonine 85, as we have heard the discussion of the importance of the mobile loop in terms of substrate binding. And so playing around with phosphorylation of these residues may play a role then in all of these aspects of enzyme activity. So I'm going to turn it over now to Celeste, who's going to talk about, well, if these are post-translational modifications of interest, how might we study these in a very directed way in our lab? So I'm going to stop sharing here and I'll let Celeste take over.
Thank you. Okay, thanks, Amy. So we've identified 3 sites that are predicted to be phosphorylated, and the question is, how do we study them? These experiments in the lab are expressing the protein from E. coli, and it's not going to have the kinases that would be involved in phosphorylating these residues. We could add a kinase, but that wouldn't be very selective, and it wouldn't really help us understand what's going on. We wouldn't be able to phosphorylate just one residue at a time. So we take an approach where we mimic the phosphorylation state by substituting in a different amino acid. So we can mimic phosphoserine or phosphothreonine with an aspartic acid or a glutamic acid. Both of these options have the same charge as the phosphorylated serine or threonine and are about the same size. And so this allows us to go in and mimic the phosphorylated state and see how that affects MDH activity. And so the questions that we're interested in are, how do these phosphorylation states affect interactions with known allosteric regulators and also the response to different pHs? So there are 6 big known allosteric regulators of MDH: ATP, AMP, citrate, glutamine, glutamate, and alpha-ketoglutamate. And we are interested in understanding what happens when you phosphorylate these residues. That will change the structure. Does that affect how the MDH responds to these regulators? In addition, we're interested in understanding how the phosphorylation state affects the response to pH. MDH is known to change its pH, known to change its activity according to pH. So at a higher pH, at pH 8, it has a higher activity than a lower pH at pH 6.4. And we know this is physiologically relevant because the pH in the mitochondria changes from 7 to 8, depending on the respiration state of the cell. So we're interested in understanding what happens when you put in these phosphomimetics. How does that affect the response to pH? So here's a typical workflow for this sort of experiment. You would start out with doing a site-directed mutagenesis, introducing what we've been doing is introducing the serine, changing it to aspartic acid at position 8 and an aspartic acid at position 45, and changing the threonine to aspartic acid at position 85. And then the standard histag purification protocol is used to isolate the protein. And then we're doing MDH activity assays, first with different inhibitors and regulators. And then we are analyzing the data. We know that now we found that these mutations lower the overall activity of the MDH protein. And so we normalize it to then see how does it affect the response to these known regulators. And if there's something that's of interest, say that abolishes the response of the known regulator, then we're doing additional experiments to try to get the KM over pH. And for the pH assays, we are looking at the MDH assay with these phosphomimetics at different pHs. And again, because the phosphomimetics lower the overall activity, we are going to normalize the data and then determine how the effective pH is affected by having the phosphomimetics. So here's some sample data. This is data that was collected by the Provost Lab. And this is looking at serine 8 aspartic acid. So, and looking at the response to alpha ketoglutarate. So with wild type protein, you have an increasing a dose-dependent response where alpha ketoglutarate is decreasing MDH activity. The more alpha ketoglutarate you have, the less activity you have. But you can see when you've swapped in the aspartic acid to mimic that phosphorylation state, this inhibition is lost. Okay, and so this indicates that this position may be responsible for some of the response to this inhibitor. So in summary, Amy and I have been working with Amy Springer and Joe, and we are looking at post-translational modifications. And these modifications can affect activity, signaling, protein-protein interactions. There are some sites that have been predicted by the mass spec data that may be relevant for cancer that we're honing in on. They have been predicted, but they have not really been further studied. And so we're testing these 3 of these. They're surface oriented and they're found in regulatory regions or part of the CS MDH interaction region. And we're looking at how they affect the response to pH and other inhibitors. I just want to say that there's lots of opportunities to study PTMs and MDH. There are dozens of PTMs that have still not been characterized. Joe's group is looking at phosphorylation sites in some of the cytosolic MDH versions, but there's still lots and lots of these phosphorylation sites that have not been characterized. And Amy and I are lucky to be working with Joe and Amy Springer on this, and it's also part of our research that we hope to publish. So this is an exciting project for us, and if you are interested in PTMs, I encourage you guys to reach out to us.